aware of that huge problem. So accessing data at different levels is not at the, in the hands of everybody. But that, in a way, is relatively simple. What is really more complex is this other direction. Okay, so how do we integrate this information into a common framework? Okay, and here is where this virtual physiological human network of excellence has tried to put some light there. Okay, this is a relatively new approach. Okay, in the 7th framework program, but in the 6th framework program, we already started doing something similar to this. In this BioBridge project that was led by Josep Roca in IDIBAPS, this is a European project also. We started building an infrastructure that would allow people to access data from different points of view, but not just to access, but also to integrate. Actually, the main goal of this project was to provide a way to produce simulations at the level of the organism, taking data from the clinical outcome of the patient. Okay, so we work on COPD, and it was successful in terms of building the structure, unsuccessful in making it real and practical. But it was just a short project. But the idea is that already at that time, this project and others already provided the tools that are needed. So my goal here is to explain you that actually the tools are known. It's just a way to understand how to put them together and how to show the results to people in a smart way. Okay? In this particular project, we basically used some software that was commercial, and we put that software connected to a central server, this BioBridge web server, that gave a transparent view to the user. So the user didn't care about what was behind, and it was just a way to, as a user that was a clinician or a patient or a researcher, to access the information in different ways. Okay? Well, this is actually what is behind the system. It's good that you don't see the text, because it's for this, the purpose is this. So the things are complex. We know that things are complex, but there is a possibility to really put them together and make some sense of them. Okay? But of course, this is just, let's say, accessing data and eventually making it available somehow. But the next step is moving from that data to actually something a step beyond. Okay, so the technology at that time didn't allow us to do what we are doing right now. But currently, what is more interesting for a researcher is not just having data. You see that the schema of this other project, this EU-ADR, which is a project that other people in my lab is involved in, it's basically the same. So it's just collecting data and making it available to the user. Okay, but what is really interesting for a biologist and for a clinician is accessing real data, real information, and visualizing that information. Okay, so in a way, not even just in this two-dimensional way. Well, actually, this is just a 3D. It's a collection of planes, so it's slightly 3D. But also even to visualize it in a way that can be useful to explain problems, to access different kinds of levels of information, etc. And here is where we started another project, which is an Avanza project, which means that is involved, a company is involved in this project. It's an industry-oriented project that is called Activa, and well, you can access this website. And basically the idea here is not just giving access to data, but giving access using a three-dimensional environment. Okay, so this is a 3D environment in which you can manipulate biological objects, and you can access actual data that is behind. Let's say a chromosome, or a set of chromosomes, or parts of the cell, or parts of the also molecular aspects of the cell, or a given tissue, different types of tissue, organs, or even the organism. Okay? If I have time at the end, I don't think that I will have time, but I can show you a video in which this system is actually used to monitor patients that have, in this particular case, cystic fibrosis. So these patients are doing some sort of interaction with mobile devices, and that interaction goes into the system, and the clinician can visualize the data using this 3D environment for them. Okay? But we'll leave it like that. Another aspect here is also that you can also visualize molecules, proteins in the system, and this allows me to introduce the actual part, the actual core of this talk. So I'm going to describe mostly what we can do right now, not just us, but in general, the people that is working in structural informatics, in a relatively easy way, or a straightforward way. Oh, thanks. Okay, and it's basically manipulating the objects that are of common use in drug discovery, for example, which are the proteins. Okay, so let me just, for those of you that are clinicians at a very high level, that you have forgotten for good what is actually a protein and the shape it has. So let's just give an extremely brief introduction of what a protein is. Okay, so the protein is not just a sequence. Okay, some of the times we go to the protein and we go to CRISPR. The protein is this thing. No, it's not like that. It's something that has a structure. It has a 3D structure that comes from this secondary and tertiary structure, and eventually the quaternary structure assembling different moieties. Okay, so from now I'm going to show you some examples in which we are developing tools to visualize structures of proteins and to see what we do with them. Okay, so it's not just having a structure of a protein, but understanding what is the function of that particular object. Okay, so this is what we're going to see from now. And let me just start by a very simple example. So let's assume that we are talking about triosephosphate isomerase. Well, you know that triosephosphate isomerase is an enzyme that actually participates in this recycling of this dihydroxyacetone phosphate into glyceraldate 3-phosphate in order to continue the glycolysis from the glucose to the pyruvate. Okay? And the interesting thing of this enzyme, which is a housekeeping enzyme, it's a central enzyme in the central metabolism, is that actually it has been shown to be involved in several cases of important diseases. For example, in Alzheimer's disease. Okay, so in Alzheimer's disease, it has been shown that it is possible, it's very likely, that nitrosative stress in the cell induces basically the formation of nitrotyrosine in this triosephosphate isomerase. Okay, that is important because it's actually a possible link between the formation of the beta amyloid plaques outside the cell and the tau fibrillation inside the cell. Okay, so that's why it's a very, very important issue and it's strongly investigated. Okay, so in this particular case, in collaboration with Paco Muñoz, they are doing experiments, not me. They realized that when you nitrotyrosinate TPI or TIM, triosephosphate isomerase, the activity of the enzyme decreases quite dramatically. Okay, so then they came to us in order to understand why. Okay, and when we realized, well, actually this molecule is like a little bit stretched, but it's pretty nice also. When you have this triosephosphate isomerase and you locate the active site of the protein, you realize that this active site can be in the active state or in the inactive state, depending on this loop. If this loop is closed, then the enzyme can produce the reaction. If the loop is open, basically what happens is that the substrate is released and the reaction doesn't occur. Okay? So, observing this region, we realized that it's actually here that we have two tyrosines that are giving hydrogen bonds that are of different types, okay, of different characteristics, depending if the loop is open or closed. Okay? So this gave us clues that this could be actually the case of what was going on here. After more experiments by the Muñoz group, they realized that actually it's true that this this TPI, is actually nitrated in Alzheimer's disease cells. Okay, don't ask me about these bands because I'm very bad explaining bands, but probably you will understand much better than myself that this is the case here. And also what they realize is that actually what happens here is that if you mutate this triosephosphate isomerase, and instead of the tyrosine, you put something else that cannot do this hydrogen bonding pattern, okay, you actually have a very similar behavior of your protein. So if you destroy the tyrosine, okay, you put just phenylalanine instead of the tyrosine, it happens that the hydrogen bonds that need to be formed are not formed. Okay, and it appears that when you nitrate the system, it's actually the same effect. Okay, so what we did there was to investigate at a very, very low level, at a very detailed level, actually the quantum mechanical level, what was the strength of the hydrogen bonding pattern that you were forming in this particular enzyme. Okay, and we investigated what happened when you have the closed state and the open state in the presence or absence of this nitro group. Okay, so investigating this, we realized that actually the nitro group makes this hydrogen bond to be weaker. If it's weaker, then you cannot form these hydrogen bonds. So the enzyme is actually opening the loop when it should not open the loop. Okay? In addition to these quantum mechanical calculations, we did molecular dynamics calculations in which we moved the system and we moved the protein, and we realized basically that the pattern of behaving of this particular hydrogen bonding network was very similar in the phenylalanine mutant that in the nitrotyrosinated, let's say, mutant, okay, modified. Okay, so which means that actually that's the case. So what happens in this enzyme is actually that those particular tyrosines are nitrated, and that makes the loop to change. But I guess that most of you say, well, what the hell is molecular dynamics? No? So let me see if this guy explains me a little bit what is that. Okay, so let me just put you an extra short primer also on what I mean by molecular dynamics. Okay, so when you have a system that, for example, you have a protein, okay, and this protein is composed by atoms. Those atoms are interacting to each other. Some of them are bond. Some of them are just interacting because one is positive and the other one is negative. Some of them are interacting with a solvent, etc., etc. Okay? So if you want to understand the system, you need something that describes the whole system. Okay? And describing the whole system means building a function like this that tells you what is the energy of each one of the snapshots that you can have of all your atoms. Okay? So a protein is not a static thing. A protein is something that is all the time vibrating. Okay? It's clashing with solvent molecules, it's clashing with other molecules, okay? So the atoms are all the time moving. Understanding what is the relationship between those atoms needs to be done by using an energy function like this, okay? Once you have an energy function like this, let's forget about the shape itself, what basically you have, it's a way to understand what is the energy of my system with respect to the coordinates that I have. So depending on the conformation of all the atoms in the system, you may have a more energetic structure or a less energetic structure, okay? In a folding process, for example, the protein is exploring this type of surface and it's finding its way to the minimum of the potential energy surface, okay? That's why folding folds. The folding process folds the protein, okay? So once we have that, then we can move the system because we can explore mathematically that surface, move on the surface, and then investigate processes, okay? This is just an example. It doesn't have anything to do with the triosephosphate isomerase, but it relates to, for example, a ring finger protein, okay? So this, you see this is a zinc ion, and these are three cysteines that are, let's say, holding this zinc atom, and this is the movement of the system, okay? So you have some water molecules that enter, that leave, you know, that are coordinated, etc., etc. So while you have this movement, you can study what is the process that occurs here. You're going to study what happens with time. So, okay, this wants to break, this wants to, you know, so you have, for example, a substrate that wants to enter in the system. It's creating some interactions, etc. Okay? That's very nice if you have the structure of the protein. If you have the structure of the protein, don't worry. You can go to the internet, you can download a molecular dynamics program, and in an afternoon, you understand how to build the system. Now Luis is going to kill me. Okay? And you can run a molecular dynamics simulation. Okay? No problem. The problem starts when you don't know what is the structure of a protein. Okay? So the structures of the proteins that are known are deposited in the database called the protein data bank, but that protein data bank is not complete at all. Okay? So it has a large number of structures, but you know, a very few fraction of the total number of structures that exist, actually, okay? So most of the time, what you have to do is to say, okay, I don't have the structure for my protein, for my Swiss prot entry, for my sequence, okay? So then, if you want to get that structure, what you're going to do is to go to a program that searches this protein data bank for the structure that is more similar to yours, okay? The sequence that is more similar to yours, okay? And then you do what is called homology modeling. You look for homologous proteins in the database, and then you look for the one that is more similar to yours. You use then that as a template, and that template is used to generate a new structure for your system. Okay? So how come you can do that? Well, you can do that because actually structure is much more conserved than sequence. Okay? So you have a lot of variation in the sequence, but not on the structure of proteins. Okay? What we're going to see now is some examples, actually perhaps even too much, let's see if we have time, of this type of experiment, okay? So having a sequence and trying to find a structure, okay? But because this is a relatively standard protocol in structural bioinformatics, let me see if I can show you. If you allow me, I'm going to show you a little video. Yeah, let's see if this works. In which I'm going to show you how we use actually that Activa Central portal that I was showing you before in order to create structures for homology modeling structures for proteins that we don't know the structure. Okay? And it's a little bit long, but I'm going to accelerate it in order to show the important parts. This is what we call the lobby of the Activa Central portal. This is just a web page, so you can go to Activa Central and use it today. And when you can enter 3 different places: the patients, clinicians, and researchers. Now I'm going to the researcher site, okay, in which you have another lobby. This is the typical laboratory for a researcher, okay. So it's probably most of you have a laboratory like this, and it's quite common actually. And then here you have several objects. So you have a microscope in which you can access those levels of biological information that I was showing you. You have calendars, you have typical things, email, etc., etc. So you have your whole desktop here. Now what is interesting here, let's see if we can accelerate this a little bit, is that at some point you can click here in the workflows. If you click in the workflows, one of the workflows that is available is actually the homology modeling workflow. Here you can enter your sequence, and then eventually you run, and the workflow runs, and you obtain your PDB file. Okay? Well, right now this is slightly not like this. Okay, so you know that these things are never like you wish it would be today. Okay, but eventually what's going to happen is that this is going to open a program that manipulates this type of protocols. Okay, I'm going to accelerate this a little bit, but basically you will see here that it will appear a workflow or a pipeline of things that have to occur. Okay, this is the important thing here. Let's see if we can see it. You enter some input. In this case, you enter this sequence. Okay, and here you see that there is a protocol that is being, it's being occurring. No? So first, this sequence is compared to the protein data bank. Then you get some templates, and then after that, the templates are manipulated in order to get the final structure. Okay. Well, let's accelerate this a little bit. What I'm showing you here is actually something extremely standard. This is not so standard. So this is a visualization of the protein in the Activa Central itself. Okay, so in the Activa Central itself, you have the possibility of visualize the protein, access, click on different parts, etc., etc. What is a good thing of this is that you don't need any extra program. So everything goes online. Okay, that's why the online title, the online word was in my title. Okay. Anyway, so let's close this. Well, as I said, what I just saw now is nothing special. Every structural bioinformatician, and we have some former master students of the best master in bioinformatics that we have in the city, actually probably the only one, and all of you know how to do these kind of things, okay? So what is interesting is that with that particular information, you can then start doing science. This is an example of a collaboration with Joan Sayos, that is in the audience, and it's the first time that he sees this structure, so I apologize for that. But this actually has been done using this protocol, the same protocol that I just showed here. So this is the example of this receptor that he's working on, and he's interested in finding structures for all the members of the family of receptors that contain this particular sequence type. Okay. Well, in this case, this modeling was relatively simple because this is an immunoglobulin fold, which is extremely conserved. You can see here that actually the degree of conservation is quite high, and this thing here means the RMSD, so the difference between one structure and the other, the difference between the template, which is the white thing, and the actual model that we have built, which is the purple one. Okay? You see that here actually the match is perfect, and this is the typical example of everything works fine. Even here, you can see that there is a region, okay, this C-terminal region that is not exactly well done, okay, but this is because this was totally automatic and this is not refined, so this is not worked a little bit more later. So you can do things automatic, but you have to be careful always, okay. In another example, we are working in understanding this, the structural basis of HLA compatibility. And for this, there are some structures of HLA, but of course there are not all the structures of all possible alleles of HLA that you can have in a system, no? in an individual. But that's extremely important because that's the basis for compatibility between donors and receptors, of, for example, of kidney transplantations. Okay, so it's very important to understand the structures of this type of molecules. And you see here that this is a typical HLA molecule in which you are presenting this peptide. You see the peptide here that the molecule is presenting. Okay, and this is the typical fold that this has because the template was pretty good. You see that the degree of conservation is also very, very high. You see that there is no gray thing anywhere except for the N-terminal and some little region here. Okay, so the structure of the template is very good in this case. And that, in this case, for example, we can use this in order to produce all the structures of all the different alleles that we have in the system in an automatic way, okay, not manual way. In another example, much more complex, we are working also in this CFTR. CFTR is it's a membrane protein that behaves as a chloride channel, and it's involved. It's the main cause of cystic fibrosis. Okay, so when you have defects on this particular membrane protein, either at the level of transcription or translation, or even maturation, then you have a problem and you have this condition. Okay, in this case, finding templates for membrane proteins is extremely difficult. Okay, because there are not so many membrane proteins that are already in the database. Okay, so you see that the degree of conservation now is much lower. These colors indicate actually that there is not a lot of conservation in general. You have changes in charges in the residues, etc. And actually the RMSD from the automatic superposition is also relatively bad. Okay, but even in this case, you can go to the system and automatically generate something. Well, we will say, well, this is absolutely a disaster, no? It is absolutely a disaster. Okay, so in this case, you cannot trust just the automatic system, and you have to really manipulate the things much more. Okay, so here is where the expertise of the structural bioinformatician behind helps you in doing the right model. Okay, so this is a right model. This is a model that is being produced now in our lab. We have tested it with some drugs, known drugs, and also we have screened virtual libraries with those drugs, and it's really promising. So okay, this is one of the parts that I showed you, that I told you before, that I have to be careful in showing in any video. Okay. Now, in general, what is the next that somebody will do when you have a structure that you are happy with? Okay. So we want to move this structure, remember, okay? So we take this structure and then we start analyzing it a little bit. No? For example, we can paint in different colors the different types of residues that we have here. So you see that the white residues are hydrophobic residues, the green are polar residues, and the blue are acidic, and the red are, or the other way around, sorry. The blue are basic and the red are acidic. It doesn't matter which is which, but what you can see is that there is a region here with hydrophobic patches, which means that actually this is a transmembrane region, okay? And actually if you add here the lipids, the lipid bilayer that should be attached, you realize that this is actually the place where the bilayer is located, okay? Not just that, it's also very important to understand that this is a chloride ion channel, no? So if you want to do a simulation of this system, you have to add the chloride ions. And if you add the chloride ions, you have to add also potassium ions, because otherwise you have a problem, no? You have an overcharged system, no? In this particular case, because it's a model and we want to ensure that it's a pore for chloride, what we do is to put actually chloride ions also in the pore. You see these balls here? These balls here are chloride ions without charge. Okay, so they are just balls that have the shape of a chloride ion, but we don't put the charge because otherwise we would be overcharging the system. Okay? So then we can put again this lipid bilayer, and then finally we fill everything with water molecules. This is actually the system that is being simulated. When you saw that thing moving before, well, that was an oversimplification of the visualization. The visualization is something like this. It may have 100,000 atoms, okay? It's extremely expensive. So sometimes you will be evaluating proposals for supercomputing power by people that is doing bioinformatics, and you will say, well, why the hell they need so much amount of time, no? So, well, because this is extremely expensive, okay? Very, very expensive. After doing the simulation here, then we can analyze some interesting results. For example, this is just a relatively short simulation. It's 30 nanoseconds for this big monster. And we have two simulations. One simulation is the wild type CFTR, okay, which behaves more or less stable, okay? And the other one is the mutation, one of the typical mutations, which is this delta 504, that is a deletion in the CFTR sequence, okay? And you see that if you delete that particular amino acid, then basically what you're saying is that the protein is not stable anymore. Actually, with this mutation, you don't even create the protein, okay? So the model is quite correct in that sense. Also, I'm not showing that, but also in terms of binding to drugs, to known drugs, okay? In another example, and this is in collaboration with Luis and with David here, we are investigating what happened with, well, actually, Luis is investigating. I'm going to explain fully the work done by Luis. Is investigating what is the way that soluble guanylate cyclase is activated by external drugs. Okay? So this is an extremely important question, and it's interesting that this being a globular protein, a soluble protein, is not crystallized in the PDB, in the protein data bank. Well, one always is suspicious that actually it may be crystallized, but in some company that are doing some research on it, but it's not released. It's still held by the company, but eventually. So we are interested in understanding what is the role of several drugs when they interact with soluble guanylate cyclase. Actually, this is a very complex problem because it's not even known if this, for example, this YC1 is activating or repressing or helping the activation, so it's still debatable. But by now, we are just interested in trying to build a model in which we can bind manually this type of molecules in order to understand what's going on. Okay? And this is what Luis has been doing. In order to validate the model that we are going to build, well, that is being built, we need to understand if it's correct or not. And if the molecule, in this case is soluble guanylate cyclase, it's cycling cyclic GMP, okay? So if it's cyclic GMP, it means that you take this GTP and you have to end up with a cyclic GMP. And to do that, you need some requirements in the active site, okay? In particular, you need some aspartics that are holding magnesium ions, they are not shown here. And you also have an arginine, the typical arginine finger in all GTPases that is actually helping the reaction to occur, okay? So actually the structure that we have here is pretty good. And this is the structure that tells us that this system is in the active form, okay? So keeping this information that we are actually with the active model, so after all this modeling process that I was showing you before, we can run again molecular dynamics simulations. This molecular dynamics is extremely long now, okay? So this is not 30 nanoseconds, this is 1 microsecond of molecular simulation, okay? So 30 nanoseconds, for your understanding, what I showed before was this region here. Okay, now we have one microsecond of simulation, okay? So this is extremely good to understand if the system is stable or not. This is state of the art, it's beyond the state of the art, I would say, for this particular type of system. And we see that actually the model that Luis built is stable. Even being stable, you see, that there are a lot of fluctuations. This is just any distance that you can imagine in the system, but more or less helps you monitoring if the system is stable or not, okay? So let's forget about that. But at some point you have some real, slightly bigger fluctuation, okay? So then what Luis did was to take a structure in this region, and a structure in this region, and try then to dock the ligands, to dock these drugs, these possible activators, in these two structures, okay? Doing that, he saw that depending on the cluster, if you use the cluster when you have 500 nanosecond simulations, or when you are in a longer step of your simulations, younger situation, well, the position of the docked molecules is totally different, okay? So this again is important, no? So when you will hear about docking and about virtual screening, about taking a structure and then binding molecules there and trying to see if they are okay or not, well, it depends on the structure. It depends on the fluctuation and the conformation that you have at that point, okay? In this case, it's clear, no, that depending on what is the frame that you use in order to dock your molecules, well, you have totally different results. What is very nice here is that in this 700 nanoseconds frame, actually the molecule, the IC1 in this case, is locating actually in a proper way. Okay. Now, why is so different? Why in the 500 nanoseconds the molecule cannot enter there and it's it's fluctuating and exploring different regions? What is the strong reason for that? Huh? When one analyzes the structure, realizes that actually the reason is a very stupid one, a very teeny one. It's just an isoleucine, this isoleucine 57, that is actually or in this position or in this other position. Okay, so depending on this, the ligand is able to enter or not. Okay, well, these are preliminary results, but we are quite happy now with the model, and now we are exploring the dynamics more now with the presence of the ligand inside. Okay, in order to see if that is stable or not. Okay, but this is the typical way to proceed. This is an extreme case. These are very long simulations and very expensive calculations. In another example, and in this case, you will allow me not to explain too much, but well, this is a molecule that is quite well known. This is I kappa B alpha. Okay, this is a molecule sorry, that participates in the NF kappa beta pathway. Okay, and our collaborators, in this case, Luis Espinosa and Ana Vigas, wanted to understand if that molecule has or not an interaction with another partner that you will allow me not to say. Okay, and they are actually right now investigating experimentally that part, and we want to see computationally if that's the case or not. Okay, and it's a very similar case. So we have to model the rest of the protein and then do dockings, and after the dockings, run molecular dynamic simulations in order to see if this is stable or not. Okay, so this is a typical, very typical procedure. Now let me jump now to the next level of complexity. So what I just explained to you now is, let's say, relatively simple. Okay, so we need to create models and then run molecular dynamic simulation. Okay, it's a relatively standard problem. Now there are problems that are slightly more complex. For example, let's think in a protein, like for example in this case RAS. Okay, and let's see if we have a way to validate if the structure is giving me slightly more information than just that it's or not homologous. If it can give me information about being active or being inactive itself. Okay? So if I can get not just the structure of a protein, but its function by observing the positions of the atoms. Okay? So this is slightly more complex. Well, first of all, let me introduce RAS for those of you that are not familiar with the inner details. So RAS is a switch. Okay? If it's on, if it's constitutively on, sometimes you have, well, most of the times you have cancers, some types of cancers. And the process of activating and inactivating is it's a very known process of exchanging GTP by GTP. Okay, so the way RAS functions is that when it's active, it's bound to GTP. Okay, and it becomes inactive because it breaks the GTP into GTP and phosphate. Okay, and it's helped by something called GTPase activating protein. Okay, this gap that allows the reaction to occur faster. Okay, basically it's a very simple system, but very efficient and works very well. What happens is that RAS itself, it's a very bad enzyme. So if it doesn't have the gap, it doesn't break the GTP. Okay, so that's the main goal here. So in principle, what we proposed here was to, let's see if we can investigate what happened if we compare structures of RAS. It's an oncogene, it's the first oncogene that was discovered, it's a well-known structure, it has a lot of structures in the PDB. So let's compare some of these structures and let's try to see if they are or not similar between them. Okay, so let's see if just by looking at the structure we realize if there are some differences that are worth exploring further. Okay, when we do this superposition of the structures, this is just the RMSD, so the distance between the structures. Okay, this tells me that basically, well, yeah, there is a value of 4 in some cases, but basically all of them are the same. Okay, if you have active or inactive, the structure doesn't give you much clue of who is active and inactive. Actually, in this order here, you will have actives, inactive, active, inactive, so nothing, no pattern. Okay, now let's see if we can do something more complex. So if instead of looking at the structure, we try to understand how this structure moves. Okay, then perhaps we understand who is active and who is inactive. So if we have a structure that is active, it's going to do some sort of movement on the active side. If it's inactive, it's going to do some other sort of movement in the active side because it wants to get the GTP, right? Okay, it wants to interchange. So then we can build another protocol. So this, forget about the rest, okay? But the important thing here is the word protocol or workflow or pipeline. Keep that in mind. In which we can analyze actually this movement. Okay? So we have a way to extract. It's a standard way. Okay? To extract movements of the system without doing molecular dynamics in a very simple way. Okay? But the interesting thing is that when we analyze the movements of all those proteins, sorry for the slide, but the important thing is the colors. These are all these proteins that I showed before, okay? And when we analyze the movements of all those proteins, we realize that, well, even if, well, you have to be very positive in looking at the plot, but you can see some differences, no? So you have some guys that have some blue colors here, which means that they are not moving in this region. The regions that are moving are basically moving more in these types and less here, etc., etc. Okay, so there is, visually even. I know that there are statisticians in the audience, so if you don't prove it with the statistics, it's not there. But even visually, you have some differences. And actually, what happens is that actually these are the inactive and these are the active ones. Okay. Now, if we move this to an analysis that clusters that information, okay, then we realize that we can actually cluster this, and we can really separate somehow the inactive from the active ones. Okay, even with this very, very simple calculation. Okay, so we have ones and the others on the side. This plot has problems, but let's advance and let's discuss them eventually later. So, so what is the message here? So we have a sequence. You have a family of proteins, okay, that have similar sequences, okay, or even not so similar, but the structures may be more similar, and the functions are even more similar than the structures. Okay, so the functions are more similar because all the proteins that are homologous with RAS are going to do the same job. Are going to open the mouth in order to get GDP and release the GDP and etc. etc. Okay, so even if the structure is not exactly the same. Okay, so can we go even a step further? Can we try to understand what is what a ligand sees when it approaches a molecule? Okay, you have a protein and this ligand is approaching what it sees. Okay, where it wants to go. Imagine that you have a protein-protein interaction. Where is it going to go? This way, this way, this way? Where exactly? Okay, so this is the problem now in structural informatics. Okay, so we don't have enough tools from what I just explained you now in order to learn that. Let's add another tool. The other tool is not just looking at the dynamics of the system, but trying to understand what is the energetics of the interactions that we have there. Okay, is this more favorable? Is this more favorable, etc. Okay. There are many groups working in this type of approaches, but I'm going to show you one particular approach in doing this type of things, which is basically analyzing the solvation of a given residue in a given place of the protein. So, okay, what is that? Okay, that means that if I have, let's say, an aspartic acid in the protein, okay, is this aspartic acid in the protein more stable or less stable than if it was outside the protein? Okay, so in other words, this aspartic that can be floating around in the solvent, okay, wants to go inside the protein or not? Is it happy inside the protein? Okay, what's going to happen is what? That the residues that are going to be part of the structure, that are going to hold the protein together, are going to be very, very happy in the structure. Okay, but not all of them are going to be very, very happy, because you will have some residues that actually participate in the active site of the protein, and those guys, they're not there to be happy. No, they are there to make a function. They are there to interact with the substrate, right? So then you will have in your protein some regions that are unstable, okay, and some inner regions that are very stable. Okay, so the inner part of the protein is going to be stable because it's happy, it's the one that is holding the structure, and other regions are not. Okay, so with this hypothesis in mind, then we can analyze our structures. For example, this is analyzing the same set of RAS proteins that we had before. Well, even if it's a little mess, okay, so this is the sequence of the protein, or at least the most important residues, you see that there are some patterns, okay? So all these guys are the same color. All these other guys are the same color. There is also a blue column here, a red column here. So something happens there. So you have a conservation of the happiness, okay, of the stability of the residues in their particular structures. This is just taking the structures and doing a very simple calculation, okay? So it's not just that the movement of the protein is conserved, it's that the happiness, the stability of each one of the residues is conserved. Okay? Then you can use this for things more complex. For example, this is an interaction between CDK2 and CyclinA. Okay? So in this interaction, you have this protein-protein interaction, and you have some surface here, and they are seeing each other, right? And then you can analyze how happy they are, but you can also analyze what is the degree of, let's say, happiness that comes to this particular residue from the others. Okay? So who is making this particular residue, more stable or more unstable? Okay? So this is an analysis of these kind of things. Okay? When you do that, at the end, you end up with things like this. This is the same molecule, but seen from two different parts. Okay? So this is the interaction region, the interface region between CDK2 and CyclinA. This is actually CDK2. Okay? Well, you see that there are positive and negative regions. These positive and negative regions refer to stable or unstable regions. Blue is stable, red is unstable. Okay? Let's have a look at this region here. So you have this region here that is red, which means that it's unstable. Okay? And this others is more stable. Okay? This is the same structure, but now when it's in the shape it has in the complex with CyclinA. Okay? So now it has changed the conformation. Okay? And now you can see that this red color is more red. Okay? So the structure had some red color there, and now suddenly changes the conformation and it's more red. So it's more unstable, which makes sense, no? Because you are modifying the structure without putting the partner, right? And now this is the final structure when you have the CDK2 inside the complex. So this red, very red color before, now it's very blue, which makes sense because that guy was expecting to interact with somebody else. Okay? So the message here is that you can take, well, the message here is that you have some residual information in the original structure that tells you, look, this is red, this is unstable. So this may be interacting with somebody else. Okay? So this is a way to find patches of interaction with other partners. Okay? In a relatively cheap way. Okay? This is extremely much cheaper than those long molecular dynamics that I showed before. So the summary of this is that when you have two proteins, let's say a catalytic protein, a catalytic moiety, and a regulatory domain or chain in a given complex, you are forming the complex, and then when you form the complex, you have the substrate entering here and the reaction occurring, right? So what I'm explaining here is that, well, you can divide this problem into two different problems. The first one is the change in shape, okay? And the second one is the real interaction, okay? And if you analyze this, then you can realize that this region here is actually the one that is going to become more stable later. So this region here can tell you something about who is the hotspot, where is the region that the protein wants to use in order to interact with other. And I can tell you that this is one of the main problems now in structural informatics, to understand how two proteins are interacting. Okay? This is another example of the application of this, what I just said. This is a collaboration with Tim Thompson in the CSIC. And in that case, they had this, this interesting pattern of interaction, non-interaction between UBC conjugating, ubiquitin conjugating proteins and ring fingers. And they realized that in some cases, some of them interact, in some other cases they do not interact. Okay? And they wanted to understand why. Okay? And eventually they come to us asking, can we recover the interaction? So if some guys are not interacting, can we recover the interaction? Okay? Well, as you can see here, this is a proteins paper. If we would be able to recover the interaction, this wouldn't be just a proteins paper. Okay? But still, okay? So the message is that we can analyze why the interactions are broken. Okay? At least we can explain who are the residues that are making the interaction to break. We cannot suggest new residues yet, but eventually this is something that could be doable. Okay? In yet another example, in this case, for example, we are analyzing, let's say, in TIM, this is triosephosphate isomerase again, all the possible, the pattern of hotspots that we can find. Okay, so these are the of hotspots that are known, and these are the residues that we detect that can be hotspots. So in principle, they match quite well. Okay, but let's go to the last part of my talk. Let's come back to this problem of the protein folding. Okay, so this simulation, this understanding of the process of folding. This is a simulation, a real simulation done using folding at home. I'm not sure if you are familiar with that. It's like this SETI at home. So you can be in your home and you can allow your computer to do calculations for these guys in Stanford. Okay, and then basically what they do is to collect the simulations done by thousands of people, and they create this long simulation here. Okay, eventually, well, this guy is going actually to a more or less stable structure, you see, and so this process works. Okay, now this takes microseconds of simulation, which means that in a, let's say, in a single computer, you can spend like 1 year running this simulation, okay? And this is just, let's say, 50 or 60 amino acids molecule, so, no? Why is that? Why is that so difficult, okay? Well, the difficulty here is that, well, we can have this thing, which is this potential energy surface that I showed you before, okay? That explains me what is the energy of the system depending on the conformation, right? But the number of paths that you can follow in this surface is exponentially big, okay? So the more radius you have, the more paths you can have, okay? And actually you have something which is called the folding funnel, which is extremely complex to achieve this point here, okay? So a simulation has this problem. You have a way to calculate this energy, but you have to explore it later, okay? So if you want to go from here to here, you can do this thing, you can go diagonally, okay? But this is like having, one of these topographic maps, these Alpina maps that we use here to go to the mountains. So you're not going to straight, no? So you're going to do something more complex, no? You're going to go valley by valley trying to reach the place. That's why it's so difficult to do molecular simulations, because you have to explore many different places. And in a way, one would expect to have something like this, something really, really soft, okay? So softening this path means, doing a lot of simulations in order to find the best path, or changing the shape of this thing. Okay, instead of having such a spiky surface, having something that is soft. If you have this surface in a softer way, then it's easier to explore. Okay, it's like having a little ball in a mountain like this. No? To do that, actually, this is done by using what are called coarse grain models for molecular simulations. Let me just introduce you this in 1 second. So coarse graining your problem means that instead of taking into account all the atoms that you have in the system, okay, why not group atoms? No? For example, if you have a methyl, if you have a methyl fragment, it's not necessary to do a simulation of the carbon and the 3 hydrogens that you have there. You can have a ball that is the methyl. Okay, so the same concept applies to bigger things. So you can take, for example, in this case, an RNA, okay? And this is the collaboration that we are doing with Janusz Winicki that we were discussing before. You can take, for example, RNA, and you can simplify this RNA by putting some balls here, okay? RNA that is really complex is actually this type of thing, okay? And when you have this, this simplification, then you just need to go to the PDB, to the protein data bank, and analyze how many of these are close to a G with this particular distance, okay? So you statistically analyze all the structures that are known, okay? And from that, you find basically some sort of simplified potentials, and you can finally get an energy function that describes each one of these very complex interactions, okay? Anyway, let's just jump this because it's just propaganda, okay? And what is interesting here is that when you are doing this simplification of your system in these little balls, at the end of the day you have to end up with something that more or less resembles the original system with all the atoms, okay? This is done by some complex molecular mechanics formula, but at the end of the day, you do not explore anymore just this coarse grain model, this soft potential, this soft potential, but you go also to the all atom potential on top of it. Okay, so you don't just explore this simple potential, but you do this multiscale simulation on the real system. Okay? I'm just skipping this part, but just to show you that actually without running extremely long and extremely expensive all atom simulations, you can reproduce what happens in those extremely long and extremely bigger simulations. Okay. Well, you can improve the model using different things. Basically, better sampling and better energy. Okay. Let me just skip this part. And let me go to the last message that I wanted to show you here. I have shown you two types of research that we do, okay, that people do. Some of them are relatively easy. Some of them are relatively routinary. Building models is relatively simple, okay, depends on the model, okay. Some of them are very complex, but there are other things that are much more complex to do. You have seen that the simple things, that simple or relatively automatizable things, okay, can be organized in pipelines, okay. So in the same way that we organize a pipeline in order to do this homology modeling protocol, okay, one can think in a pipeline that describes, for example, DNA-seq experiments, okay, this is more familiar with what you do here in the Biorun, or RNA-seq experiments, for example, okay. So you have the tools to describe complex things with protocols that are reproducible, okay? And that's where we decided, and so sorry for the commercial here, but in a way to move all this part of protocols, all this part of standard ways of doing things into what we call a seria, which is a separate part. So in a way, you can distinguish in a research group what you do when you do routine or when you are not doing routine. And I think it's good that every research group is able to distinguish these two parts and create protocols, automatic protocols that informatics allows to do that in order to solve complex problems in a routinary way. Well, anyway, I want to finish my talk by thanking many people, okay? Basically my group, my current group right now. I think that I saw Maria Angeles somewhere entering. Yeah, she's there. She's hiding, but she's there. And the rest of the people that most of you have already seen her faces during the talk. Also, people that has been in the group in the past, and the collaborators in this O2H link with which we are doing this Activa Central portal, and the people that collaborated in the BioBridge group. But of course, for me, what is more important is the collaborations that we have with experimentalists, because on the one side, we can do a lot of research developing new methods, but if those methods are not used in a real setup, it's quite useless to develop things. No? So thanks to all these people that is here, most of them are experimentalists, and some are here actually. And thank you for your attention, because at this time, this is not easy to do. Okay?